So my name is Andrea Serio, and I am a reader in neural tissue engineering here at King's, and one of the group leaders at the UK DRI Center. My research focuses on bringing together engineering, neuroscience, neurobiology, and neurodegeneration in order to make more complex, accessible models of the nervous system, in order to understand how our brain and spinal cord are wired together, or more to the point, how they stop being correctly wired together when diseases such as Alzheimer's or ALS occur. Here at the UK DRI at King's, we've set up what we call a Fab Lab, which is a fabrication facility for biology to give neurobiologists and clinical scientists everything that they need to design what we call lab-on-chip devices. Small microfactories of cells that would convince your otherwise conventional cell culture to have interesting and complex properties. Either wire a circuit in a certain way to mimic the brain or the spinal cord, or give you the power to provide minute quantities of a drug exactly where you want it, and in other words, having more complex and refined ways to interact with your biology while you are in the lab. And so when I opened my lab, one of the first questions we had was, does it matter that we are trying to study these cells at much shorter scales than they are in the body? And so we wanted a system that allowed us to do a couple of things. Control how long these cells are in a systematic way, being able to visualize one single axon, which is this long connection, within this complex culture. We design a system that has micro grooves, little channels that will convince the neurons to align in a certain direction. We engineered the surface of this substrate to be very cell-friendly in a specific way that would convince the cells that they would find themselves in the specific position during development where they are looking for a muscle to connect to. And with a couple of other engineering tricks, we basically made a system that allows us to have the same cells at micron, millimeter, and centimeter scale. And lo and behold, we discovered something that nobody's noticed before, that length has a lot to say about how these cells develop. And there are special characteristics in the way that these cells maintain their biology at a distance that only get activated when they reach a specific length. In other words, there's a specific physics-based switch rather than biology-based that we wouldn't have noticed if we didn't engineer the environment. Basically, we have designed, built, and optimized a lab for a biological question or an experiment that might be the key thing that either us or another biology lab might have. And we can share freely both the ideas, the designs, and the actual physical devices. And we find that very stimulating, and we believe it's going to give therapies and drugs and discoveries much faster and better.